Hello, hi, a very happy good evening. So this is Krishna Ma'am. We're talking about the story of life on earth, and that's biology. So thank you so much for joining my lecture today. So we are here talking about Mendelian genetics. So we are two-way through the chapter. So we're already done talking about part 1 and part 2 of Mendelian genetics. We also solved a lot of problems. So I guess that helped you understand the basics. So today we are going to build on to this. So this is part 3 of Mendelian genetics. So we are going to move forward. So what is chromosomal theory of inheritance? What are pleiotropy? What is polygenic inheritance? So what are mutations? That is all what we are going to talk today. So this is basically part 3. And yeah, so here it says Mendelian genetics as well as some questions. Ma'am, are we going to discuss some questions? Hi Nishra, yes, a very happy good evening. So thank you so much for joining my lecture today. So please do like, share and subscribe. Yeah. So on the screen it's written Mendelian genetics, within brackets is mentioned questions. So are we going to solve questions today? Yes. So Nishrat, I'm addressing your question today. Well, so at the end of the slides, so at the end of the theory session, so I have almost 15 questions attached. So it will be homework because they are time-taking questions. So you will solve all the 15 questions that is attached at the end of this lecture, and come on Wednesday I'll be discussing these questions. Okay, yes. So today I will teach you so that we are almost done with the chapter. So Wednesday we will discuss these questions and then we move on to pedigree analysis. So this week we are going to have a Menti. Okay. Ma'am, when will Menti quiz happen? So Menti quiz will probably happen on Friday. Okay, Nishra? Yes. So we are going to have a Menti in human physiology. So I guess I have made it clear. So in this lecture, so I hope you guys are able to get access to my PPTs, for which you have to download _Point app. So you get my PPTs in the form of PDF. So open today's PPT in the form of PDF, and at the end you will find 15 questions from genetics, the topics that I have already discussed with you. So please solve them and come on Wednesday's class. I will discuss them for you. Right? So we quickly go forward with the theory because I guess I'm just skimming through the theory and it is pretty easy. Right? So shall we move forward? Yes, Nishrag. Okay, so I hope Nishrag you had a very good weekend and you've been solving good amount of questions. Yes. So let's see what is today's agenda. So what are the things that we are going to talk today? So we shall discuss about chromosomal theory of inheritance. So we are moving forward. So we stopped with dihybrid cross. So after dihybrid cross, we solved 10 questions, and then today we are moving forward. So the next topic in line is chromosomal theory of linkage. Then you have genetic linkage. Sorry, you have chromosomal theory of inheritance, then you have genetic linkage, and the next one is polygenic inheritance, and the last one is pleiotropy. So these are the topics that I want to address today. So let's see how much I can finish. So let's first talk about a chromosomal theory of inheritance. So long we were talking about how the father of genetics, 𝐕𝐻 Mendel, went forward with his work. So he basically spoke about the inheritance of certain things, or what he named as discrete factors, move from one generation to the other, that is from the parental generation to the offspring. So today we know that discrete factors are called as genes, right? So though he did a lot of work, though he did a lot of analysis, but his work was not recognized immediately. So sometimes it happens, right? You work really hard, but you don't get a recognition, you don't get a result, right? So you have one beautiful example of 𝐕𝐻 Mendel. So he did a lot of work. So he was the first one to think in a very different way, without which today we would not have this genetic era, right? But his work was not recognized for a very long time. Why so? Why wasn't his work being recognized? Yes, Prince, a very happy good evening. So thank you so much for joining my lecture today. So please do like, share and subscribe. Yeah. So why wasn't 𝐕𝐻 Mendel's work recognized? So that is because, number one, there was not a lot of easy access to communication in that period. So he worked towards the 18th to the 19th century, and communication was a big task in those times. So just like today, if I want to talk to someone across the continent, it's pretty easy. I just have to drop a mail, I just have to do a WhatsApp, or I just have to do a call, and it's very simple. It's even simple with WhatsApp call, right? But in those times, it was pretty difficult to communicate from across the continents, across seas, to talk to a scientific community, right? So that was one reason Mendel's work was not recognized. Number two, his concept of genes, his concept of this getting passed over, and his concept of bringing this Punnett square, which is a statistical analysis, into biology was not accepted. So what is more interesting is today we call 𝐕𝐻 Mendel as father of genetics, but he was not a biologist, he was a mathematician. So the biologist people were like shocked. So how can you bring mathematics into biology? So we take biology because we are scared of maths. Well, so that is a myth. So no bio student is scared of maths. So without maths, no genetics is possible, right? So this is some basics that I'm showing you where you need to apply maths. So in complex genetics, you definitely need maths, right? So people couldn't initially accept him bringing mathematics into genetics. So that was reason number 2. And number 3, he said something is getting passed on from one generation to the other generation, right? So from parental to the offspring. But he didn't have the proof to show this is the gene and this is getting passed on. Yes, he did experiments. So I also will do experiments and say, so there were creatures who were as tall as you, who were twice the tall as you, who lived in the ancient era. But unless and until I show you the skeleton of a dinosaur or the image of a dinosaur, you guys have no reason to believe what I'm saying, right? So I can be even bluffing about what I'm saying, but you guys need a solid evidence. So science is about having an evidence. So without evidence, the community fails to believe or does not want to believe you. So though Mendel did a lot of experiments and he concluded, so he was not able to prove it in a physical way. So physically he did not have an evidence to show people, but theoretically he had a lot of evidence and experimental proof, right? So that was also the reason Mendel's work was not recognized until a very late time. So very later his work came to be recognized, and his work was slowly recognized by a group of people. Yes ma'am, a very happy good evening. So thank you so much for joining us today. So please do like, share and subscribe. So his work was recognized. So when his work was recognized, so that time the microscope was already discovered. So with the help of the invention of the microscope, so people were able to see the cell very clearly, right? So when they were able to study the cell clearly, they were also able to study how the genes are getting passed on from a mother cell to the daughter cell. So now they understand, they understood, they verified, they authorized, they acknowledged Mendel's concept. Okay, so that is how his work came to be re-established or re-recognized years back. So this is also one defining, standing example of how hard work never fails. So hard work might take time to prove itself. So good work always takes time to prove itself, but good things always take time. And once the time has crossed, you're always in the successful position. So always don't fall down. So I am reading so much, but I don't get good results, I don't get a good output. So please wait. So things take time in its own way. So everything has to fall in place at the right time, and George Mendel was one brilliant example of it. So okay, so you have asked me a doubt in the Telegram. Okay, I'll get back to you. I'm really sorry. I'm packed with classes, so I don't get time to get a reply to you on Telegram. So after class, or maybe by the end of today, I'll get back to you on Telegram. Okay, Prince? Yes? Right, so what is chromosomal theory of inheritance? So let's move forward. So Sutton and Boveri, so these were the two scientists who came up in the next era after George Mendel. And they said pairing and separation of a pair of chromosomes would lead to the segregation of a pair of characters they carried. So when I say here a pair of factors, here I mean that a pair of factor, a pair of character refers to genes. So on the chromosome we have genes, right? So your genes are present in the DNA, and your DNA is too big to compress into a nucleus, right? And hence your nucleus are organized into structures called as chromosomes. So when a chromosome segregates, obviously the genes that are present in the chromosomes would also segregate, right? That is what is called as chromosomal theory of inheritance. Fine. So chromosomal segregation plus Mendelian's principles of inheritance of one gene and two genes is what gave birth to the chromosomal theory of inheritance. So the experimental verification, so here they understood now if there is no verification, no one is going to believe our work, right? So me continuously talking without solving problems will do no good to you. So only when I solve questions and only when I clear your doubts, you know, okay, so ma'am knows something that she's talking about, right? So much in the same way, we all need evidence, right? So here experimental verification was done by Thomas Hunt Morgan, and he did this experiment in fruit fly. The fruit fly is called as Drosophila melanogaster. So the fruit fly is again very easy to cultivate, very easy to study. It's a very good model. So this is how your chromosomal theory of inheritance came into picture. So these are your house flies. So now let's move forward. So the theory, this chromosomal theory of inheritance was based on the following observations. So what are the observations? Let's check now, right? So number one, Homologous chromosomes migrate as discrete structures independently. So, okay, so first let me put in all the points so that it's easy for us. Right. So, we all have 23 pairs of chromosomes, right? So, together we have 46 chromosomes. So, why do we say we have 23 pairs of chromosomes is because we have one copy coming from the mother and one copy coming from the father. So, if I say we have 46 chromosomes, so let's talk about chromosome 1. So, we have two copies of chromosome 1, one coming from a father, one coming from a mother, right? So, those chromosomes, both are chromosome 1 and it is a pair, right? So, those chromosomes are called as homologous chromosome. So, homologous chromosome pairs migrate as discrete character structures independently. So, when you have one copy from dad and one copy from the mother, but at the time of gamete formation, only one copy comes, right? So, gametes are haploid and they have only one copy of the pair of chromosomes that you possess. That is the reason. So, that is your theory 1. So, this is the first observation, sorry. So, sorting of chromosomes appear to be random. So, I said in your gamete, only one of the pair comes into picture. So, which one pair comes is pretty random. So, you do not know. So, that is completely random. So, point number 3, each parent synthesizes gametes. So, they contain only half of their chromosome complement, which is already explained. Equal genetic contribution from each parent. So, equally the parents contribute to the genetic information. So, what you are today, your height, your features, everything is the equal contribution from both your parents. It is not that only one parent is given more advantage. No, the gene in that parent might be dominant, but the contribution is equal from both the parents. The offspring with the same chromosome number as that of the parents are produced. Though the gamete has only one of the pair of the chromosomes that you possess, but the offspring, the zygote, will have twice or the same number as the parent, right? So, this is the observation, or these are the conclusions of your chromosomal theory of inheritance. So, let's move forward. So, now what is linkage and recombination? So, now I have made it clear the genes are present in the DNA and the DNA is condensed into structures called as chromosomes, right? So, suppose, let me draw a chromosome. So, this is a chromosome. Suppose here I have a gene and here I have a gene. So, these two genes are very closely located, right? They are not very far apart. They are pretty closely located. Physical association of genes on a chromosome is called as linkage. So, the association of genes, the physical distance of genes on the chromosome is called as linkage. So, when they are very close by, we call it as complete linkage. So, when the genes are physically very close by to each other, so that is called as complete linkage, okay. So, generation of non-parental gene combinations are called as recombinations. So, if you have ever read the chapter in class 11, chapter number 10, cell cycle and cell division, so which talks about how in the process of meiosis crossing over happens. So, you are not exactly like your mother or your father, but you are a combination of two, right? Because there happens an exchange or crossover of information from both the chromosomes. So, suppose say I have one set of homologous chromosomes. So, this is chromosome number 6 and this is from mother, this is from father, okay. So, sometimes there is crossing over of information in both the chromosomes. So, the information crosses over like this, right. So, that is why you are a hybrid, you are a recombination, you are a different version or you are a combination of both of them, right. So, now the newly produced chromosomes will be like this. So, here the information is crossed over. Now, this can also be in your gamete, this can also be in your gamete, right. So, when the genes are very closely linked, the possibility of recombination or a new combination is very less. So, suppose if this region crossovers with another chromosome like this, so both will cross over together, right, because they are very closely linked. So, the possibility to have a new combination is pretty less here, right. So, suppose if I have a chromosome and the genes are pretty apart, So I have one gene here, I have another gene here. Then during crossover, the genes can separate from one chromosome to the other. So when the genes are pretty apart from each other, this is called as incomplete linkage, and the number of recombinants or the number of non-parental combinations can be greater in a incomplete linkage. Okay, so that is what I mean by linkage and recombination. So Mendel could not talk about linkage because he focused on law of independent assortment. He took up genes, or he worked on characters that were present on the same chromosome very, very close by. Okay, so that is the reason he could not work, or he could not even think of linkage, and this was later done. So some genes are tightly linked, so they show less recombination, because if this is recombined, both will move to the different chromosome, right? So there is no probability that it can give you new combinations. Fine. So some genes are loosely linked, so you get high recombinations because they are physically a long distance apart. Fine. So yeah, so more closely linked genes and less closely linked genes. So more closely linked genes are called as complete linkage, and less closely linked genes are called as incomplete linkage. So linked genes or complete linkage will have more parental combinations and less of recombinants, and this is vice versa for incomplete linkage. So he was Thomas Hunt Morgan who spoke about linkage. So here we have an example in your textbook for tightly linked genes. So you don't have to do or understand the crossing, but just understand. So we are talking about Drosophila, a fly. So we are talking about two characters here, brown body and red-eyed female. The body is brown and eye color is red, right? This is your normal type. This is your female, the wild type that is prevailing in the population. And in the male, you have yellow-bodied and white eye, brown-bodied red eye, yellow-bodied white eye. Okay, so you can see the genes for the body color and eye color are located very close by, right? So here the parental combination is pretty high, that is 98.7%. So this is complete linkage, right? But the recombinant type is very rare, 1.3%. So hardly one offspring out of the 40-50 offsprings the fly gives birth to will be of the recombinant type. So I never said here you will not have recombinants, but the amount of recombinants will be less. The amount of parental combinations will be pretty high. Okay. So let's move forward. Okay, now what is polygenic inheritance? So we are going to talk about polygenic inheritance. So poly means many, right? Genic means genes. So many genes affect a particular character or affect a particular trait. So that is polygenic inheritance. It is pretty easy. So students always get confused, or they swap the difference between polygenic and pleiotropy. But it's very easy to understand the essence if only you understand how logically you are speaking about. Polygenic, poly is many, genic is genes. So many genes affect one character, so that is polygenic inheritance. So traits controlled by more than 3 genes is polygenic. Okay, so they involve the influence of the environment as well, because sometimes the environment is very different. It behaves very, very roughly, right? So now you have unpredictable rain. So recently 3-4 weeks ago, so you had heavy rains. So this is not a rainy season, this is winter season, right? So the climate is so unpredictable. So we have greenhouse gases, we have global warming, you have El Nino effect. So everything's happening at the same time, and we don't know where we are going to go and end, right? So we all know that we are destroying Mother Earth. So we all know we have destroyed it so much that the repair will take a lot of time, a pretty lot of time. But still we continue to move forward, right? So that is how smart these human beings are called. Well, so who cares? So today we have bread and butter, that's all we care for, right? Yeah. So example is human skin color. So in the human skin color, so this human skin color, so for this color of my skin is I'm neither very fair, I'm neither very dark, right? So this skin color is coded by 3 genes. The genes are called as A, B, C. So there are 3 genes which codes for the skin color A, B, C. So each gene has a pair of allele, right? So if I talk about gene A, so it can be like this. So it has a pair of alleles with it. So if I talk about gene B, it has a pair of alleles with it. If I talk about gene C, it has a pair of alleles with it, right? So in this trait, the phenotype reflects the contribution of each allele. The effect of each gene is addictive. So there are 3 genes, and if everything is in the dominant condition, the person's skin color is dominant. If everything is in the recessive condition, the person's skin color is pretty light. So when it is in the heterozygous condition, it is in the intermediate color. So each type of allele in the genotype determines the darkness or the lightness in the skin, right? So when all the genes are in the dominant condition, the person is very fair, very dark. The more amount of pigment is being produced, right? If there is less amount of pigment, that is if everything is in the recessive, that is light color. If it's in the intermediate, so it is the intermediate, if it's in the heterozygous condition, it is the intermediate. So I guess I have an intermediate gene because I am neither too fair, I am neither too dark. So that is how you see how a single character is controlled or governed or harnessed by 3 genes. Don't confuse this with blood grouping. In blood grouping, you have 1 gene and many alleles. 1 gene has 3 alleles. Here, many genes work on a particular character. So, your blood grouping is codominance and it is multiple allelism, whereas this is polygenic inheritance. Done? Fine. So, let's move forward and let's talk about what is pleiotropy. Polygenic is many genes, they affect or they have an effect on a single phenotypic expression. Whereas pleiotropy is where 1 gene, so here you have 1 gene affecting many phenotypes. So polygenic is many genes, 1 phenotype, 1 character. Now in pleiotropy, you have only 1 gene, but if you do not have the gene, a lot of phenotypic expression can be affected. It is vice versa. Polygenic, many genes, 1 character. Pleiotropic, 1 gene, many phenotypes. Okay, so that is how you correlate. A single gene can exhibit multiple phenotypic expressions, so that is a pleiotropic gene. So 1 gene is enough. If 1 gene is absent, you have a lot of problem, right? So that comes under your pleiotropic gene. The effect of gene on the metabolic pathways can contribute to different phenotypes. For example, we have a lot of chemical reactions happening inside our body right now. So for example, say if you had roti and dal in the afternoon, so now your body is completely into digesting the food, right? So probably if your metabolism is quick, your digestion would have been done. If it's pretty slow, it is still happening, right? So let's say it is like that. Okay, so it's moving on its own. Oh, why? Relax, relax. Yeah. Fine. It's moving in reverse on its own. Right. So I guess the panel is pretty sleepy after lunch. Yeah, so we are here. Right, so you have a lot of metabolic activities happening inside your body. So you have a lot of pathways, right? So if I had rice in the lunch, so the rice is broken down into the form that is understandable by the body. So that is the entire process of digestion. So the rice that I eat is broken down into starch and starch into glucose, because the end product that my body can utilize is glucose. Then that glucose is utilized in the process of making cholesterol, in the process of making my nucleotides, in the process of making enzymes, a lot of other things, right? So if a single gene is not there, it can lead to a lot of disruption in your metabolic pathways. So one example we'll talk about is phenylketonuria. So phenylketonuria is an inborn disorder. So for example, you have different amino acids, if you've again read the topic biomolecules, right? So if you have a list of essential amino acids and you have a list of non-essential amino acids. So suppose if you have phenylalanine, so this phenylalanine amino acid in your body has to be converted into tyrosine. So this conversion is catalyzed by an enzyme, that is phenylalanine hydroxylase. So if this enzyme is absent, this conversion does not happen. So then this phenylalanine accumulates and it leads to a condition that is called as phenylketonuria. right? So now if this enzyme, the gene that is coding for this enzyme, phenylalanine hydroxylase, is absent, so then it leads to a lot of condition. So mutation in this gene can lead to mental retardation, can lead to reduced metabolism, a lot of functions like this. So one gene can lead to a lot of phenotypic effect. So if you Google the pictures of people who are suffering with phenylketonuria, from the face itself you can identify this person is not normal. So one gene can make a lot of difference as well, right? So that is pleiotropic inheritance. Okay, so we move forward. Sex determination. So what are we going to talk about? Sex determination. So I guess this is the last topic for this evening. So sex determination. So who determines the offspring that's going to be born, is male or female? So today we know how it is happening, but years before it was a mystic miracle, right? So it was a puzzle which we didn't know. Who decides it? Who controls it? So we didn't know so much. So scientists have been continuously working on what determines the sex of an offspring. But the initial clue of the sex determination came from insects. So how did they find out in insects also how the sex is determined? So basically a lot of things you understand when you understand the cytology of the cell. When you understand how the cell works. The cell is the very complex, the smallest, the complex thing that you can find on earth, right? So cytological observation, cyto is cell, so cytological observation in insects, that gave us the basis for sex determination. So there was one more scientist here, he is known as Henking. So he traced a nuclear structure all through the spermatogenesis in the few insects. So spermatogenesis is the process of production of sperms in the male, right? So now spermatogenesis is the process of production of sperms, which is the gamete. So if the insect is diploid, if the insect has 46 chromosomes or 23 pairs, so after spermatogenesis the sperm will have only 23. Right? So when he went on to trace the structure of the sperms, then that gave us a clue as to what is exactly happening during the sex determination. So 50% of the sperm received the structure and the rest did not receive because it's going to be haploid, right? So X body, so that is X chromosome. So he understood, so your sperm is XY. Right? So 50% had the male gene and 50% was X gene. So later he found out the shape of your X chromosome and it looked like an X-shaped structure. So hence it came to be known as the X chromosome. Alright? So that is known as the sex chromosome. So how is the sex determined in a lot of cases? So in humans, you know it is the XY type, right? So the male is XY in humans and the female is XX. So the gametes can be X or Y and here the gametes can be only X. So XX combination makes a female and XY combination makes a male. So now who determines the sex here is the male, because the male have two different types of gametes. That is, the male has heterozygous gametes. So this is heterozygous, right? 1X, 1Y. So this is heterozygous and XX is homozygous. So since male have your heterozygous condition, so males determine the sex in your female. And this type of sex determination where the males determine, it is called as male heterogamety. So here insects in Drosophila, they have XO type. So the females have XX and the males have XO. So again by the fusion, if you have XX, it is female, XO, it is male. So since the male chromosome or the male sex chromosome is heterozygous, the sex is determined by the male. So in case of your Drosophila, in case of your insects, in case of your humans, the sex is determined by your male. So this condition is known as male heterogamety. But in birds, it's altogether different condition. In birds, the male is homozygous, the female is heterozygous. So this is known as female heterozygosity or female heterogamety, where the female determines the sex of the offspring, right? Fine. So let's move. So now you have something called as dosage compensation. So it is pretty complicated because it is being used in a present study, a present field of study that is called as epigenetics. So what is it? Okay, fine. So before we go into complex terms like epigenetics, so like genetics, you also have a field that is called as epigenetics. Okay. So now what is this dosage compensation? So in case of your human male, you have XY. In case of your human female, you have XX. So here the male has only one X and the females have two X, right? So now, since to manage or since to equalize the number of functional X chromosomes, one X chromosome in female is always silenced. One X chromosome is silenced toward the life of a female. So this silenced X chromosome is called as your bar body. So all females have two X chromosomes, or we have a pair of X chromosomes, but since male have only one X chromosome, and if we have two X functional chromosome, then we'll have twice the function. To equalize the function, to equalize the work in both male and female, one X chromosome automatically silences itself, and this is your bar body. This is towards the periphery of your nucleus and your functional X chromosome is towards the center. Alright, so this is called as dosage compensation. Hi Natasha, hello, hi. A very happy good evening. So thank you so much for joining my lecture today. So please do like, share and subscribe. Right. So in case of insects, the male has XO and the female has XX. Now how does this work? So here also, does the X silence itself? No. So this increases its capability and works twice. It works twice the level here. Alright. So that is how it happens. So it's a very beautiful way of how things work in nature. And we still haven't found or discovered the entire way how things work. We're still in the process of learning because nature has a lot of things to tell us. So the last slide, okay, so we also have sex determination in honeybees. In honeybees, it's a different case. So always you have an exception, you have a different case, a unique case, right? Honeybee is one of them. So in honeybee, the males are formed by a different way and the females are formed by a different way. So in honeybees, the females are diploid and the males are haploid. So in a honeybee population or in a honeybee community, you have three types of people, right? So queen, you have worker and you have a drone. So queen and worker are female, so they are diploid, and the drone is a male and that is haploid. So if there is a male and a female gamete, it always fuses to form a queen or a worker, right? Say for example, this comes from this, right? So who produces this N? The male. So the males undergo mitotic division to produce haploid gametes and hence they form the male gamete, right? So this type of system is called as the haplodiploid system. If the egg is unfertilized, it becomes a drone. Okay, so this is parthenogenic, you can say. Done? So let's move forward. So what are mutations? So sudden change in the DNA or the base sequence in your DNA is called as mutations. So you have your DNA. So your DNA is made up of nucleotides. For example, the nucleotides we have are A, G, T, C, etc. So if there is a sudden change in this, suppose if G is deleted. So instead of G, if I have GC, TC, that is a small change, right? So this leads to a change in the phenotype, and this sudden change in the DNA sequence is known as your mutation. The phenomena which results in the alteration of DNA sequences and consequently results in the changes in the genotype and phenotype of an organism. So this is defined, or this is called as mutation. So mutations can be deleted. So here G is deleted. Insertions, instead of G alone, C is also inserted. Duplications, right? So ATGC, so it can also be AG, AGTC. So duplications are also there. So this leads to the number of disorders in the chromosomal number. Sometimes you can have one chromosome less, sometimes you can have one chromosome more. So mutations are pretty common in your cancer cells because uncontrolled growth leads to mutations. So one type of point mutation, when there is a change in a small base pair, that is point mutation. So what is frame shift mutation? We will see in your molecular genetics, not now. So these are the different types of mutations that we have. Okay, so for point mutation, you will remember sickle cell anemia. When there's a change in a single point in your DNA, that is point mutation. So with this, we are done. So as promised, I have a lot of homework problems here. So you can solve all these homework problems on Wednesday. I'll be solving these. right? So I am attaching these questions so that you can download my PPT and you can solve them and come. The questions are pretty interesting and it should not take more than 20 minutes for you to solve the entire lot. It's very, very easy. And on Wednesday in the live class, I will also solve so that you can cross-check how you have solved and what is the right answer. Alright? Yes? So I hope you guys understood my way of teaching and please do solve the questions. So thank you so much once again for joining my lecture. So please do lend us your support. So let your friends also know about our channel. So please once again don't forget to like, share and subscribe. So I'll meet you on Wednesday at 4 o'clock. So until then, this is me signing off. So take care and bye.